It was a night just like any other when Tomas realized that something was fundamentally wrong with his eyes. He was taking a routine stroll along the very same paths near his village that he had walked thousands of times before with his friends. There were no streetlights to guide them, and the trails were illuminated solely by the soft and steady glow of the distant moonlight. This had never been a problem for him in the past, but suddenly he realized that he could no longer clearly make out the edges of the path under his feet. He stopped and hesitated, while trying to find a single reference point to anchor his vision, but the edges of his world were blurring into a confusing and dark haze. Without knowing it at the time, Tomas was experiencing the very first biological symptom of a condition called retinitis pigmentosa, which is commonly known as night blindness. This silent and invisible shift in his sensory perception marks the start of a journey that affects millions of people across the globe, and it represents one of the most intense psychological and medical challenges in the field of modern biology. The story of Tomas is not just a personal tragedy, but is the starting point for a massive scientific race against time to understand how our eyes process the world and how we can repair them when our genetics fail us. To understand why the lights begin to fade for some individuals, we have to explore the incredible neurosensory tissue known as the retina, which lines the very back of the human eye. The retina is actually an extension of your central nervous system that develops during the earliest stages of life, and it functions like a highly sophisticated sensor that catches particles of light called photons. It is organized into distinct and perfectly arranged layers of neurons that are interconnected to process visual information before sending it to the brain. At the very edge of this structure sit two primary types of photoreceptor cells, known as rods and cones. These cells are the fundamental engines of our sight, and they are so sensitive that they can be triggered by the impact of a single photon. In a healthy retina, there are approximately 120 million rods and around 7 million cones, and the delicate harmony of these cells is what allows you to see both the vibrant colors of a summer flower and the subtle movement of a shadow in a dark hallway. The rods are the specialized cells that allow us to function in low-light environments because they are specifically designed to respond to low photon intensity. Inside these rods is a light-sensitive molecule called rhodopsin, which acts as the chemical trigger for vision in the dark. However, the biology of the rod comes with a unique trade-off because these cells cannot perceive color and can only provide a view of the world in black and white. On the other hand, the cones are packed with proteins called opsins that only respond to high photon intensity. These are the cells that give us our sharpest details and our ability to see a full spectrum of colors, but they require a significant amount of light to function at their best. Most of our cones are concentrated in a central area called the macula, which provides us with visual acuity, while the rods are spread throughout the rest of the retina to give us our essential peripheral vision. When a person has retinitis pigmentosa, the genetic instructions for these rod cells have been mutated, which causes them to become damaged and eventually stop functioning altogether. In a tragic process known as programmed cell death, these cells essentially shut themselves down because they can no longer maintain their internal stability. This loss usually begins at the outer edges of the retina and moves inward over many years, which is why it often goes unnoticed until the damage is severe. As the rod cells die off, the patient begins to experience what is known as tunnel vision, where they can still read a book or recognize a face because their central cones are healthy, but they find it impossible to navigate a dark room or see objects in their periphery. Eventually, the death of the rods creates a toxic environment that also kills the central cones, leading to a total and permanent loss of sight. While retinitis pigmentosa is the most common form of hereditary blindness, it is not the only way that genetic errors can disrupt our connection to the world. Some conditions appear much earlier in life, such as Leber congenital amaurosis, which can cause severe vision loss in infants, or achromatopsia, which leaves a person unable to see any color at all, making the entire world appear in various shades of gray. In other disorders like Stargardt disease, the opposite happens because the mutations specifically target the cones and the macula first. This creates a situation where a patient can see perfectly well in low light using their rods, but they find it impossible to read a street sign or see the fine details of a human face. These variations show just how many different genetic keys must function perfectly for us to maintain our visual health, and why researchers are so focused on finding a universal solution. At this exact moment in history, there are no approved treatments that can completely stop the progression of these diseases. But biotechnology is providing a path toward incredible hope. To design effective therapies, scientists must conduct foundational research into the biochemical pathways that are disrupted when a mutation occurs. This is where medical technology has made incredible leaps by allowing researchers to analyze disease models in ways that were once considered impossible. Scientists are now creating what they call avatar mice, which are genetically modified to carry the exact same condition as a human patient. Even more revolutionary is the development of human retinal organoids, which are tiny versions of a human retina grown in a laboratory from a person's own cells. These organoids allow researchers to test new drugs and genetic interventions without ever putting a human volunteer at risk. There are currently two major paths that the scientific community is following to treat these rare conditions, and the first is known as precision gene therapy. This involves delivering a functional copy of a mutated gene directly into the retinal cells to restore their survival and function. A primary example is a treatment called Luxturna, which has already restored vision for many patients with a specific gene mutation. The second path involves what are called agnostic therapies, which are designed to support the survival of photoreceptor cells, regardless of which specific mutation is causing the issue. The goal here is to provide the cells with the protective factors they need to resist cell death and stay functional for as long as possible, which could potentially help millions of people like Tomas who are still waiting for a cure. As we look toward the future, the search for new treatments in the field of ophthalmology is a powerful reminder of our capacity for innovation and the resilience of the human spirit. Every breakthrough in the laboratory brings us one step closer to a world where a late-night walk in a village is a source of joy rather than a source of fear. Protecting your vision and understanding the science behind how you see the world is a vital part of taking care of your long-term health and wellness. If you find the intersection of genetics and biotechnology to be as important as we do, make sure to keep exploring the latest medical findings and breakthroughs that are transforming our understanding of the human body. Staying informed is the best way to support the scientific community and stay prepared for the future of healthcare. Respecting the complexity of our own biology and the researchers working to repair it is a step toward a reality where light always wins. Your vision is a precious window to the world, and knowing the science that keeps that window open is the first step toward valuing it for a lifetime. Keep watching and stay curious about the miracles of modern medicine, because the next big discovery might change everything for someone you love.